Hello students, in previous part we have studied about the different experiment proposed by different scientists such as Griffith. Griffith showed the transformational experiment, and after that re-experiment of Griffith, Avery, McLeod and McCarthy, which is known as biosynthesis chemical experiment, and after that the confirmatory experiment proposed by Hershey and Chase. It was shown that DNA is the genetic material. Now in this part we will discuss the differences between DNA and RNA, the genetic material. DNA and RNA. What are the difference between the DNA and RNA? Why is the DNA genetic material not RNA? What are the basis of genetic material between the differences between the DNA and RNA? As we have seen that DNA is the genetic material which was proposed by Hershey and Chase. Now we will differentiate between DNA and RNA. First one is the genetic material which show mutation, replication. What are the different characteristics of genetic material? It should be replication, it shows stable mutations, the expression of the information may be transferred the normal way. That should be the genetic material. Now in this part we will discuss on difference between DNA and RNA on the basis of replication. Why is the DNA genetic material not RNA? We will differentiate between this one on the basis of replication. Replication means the element which has the ability to make its copy. It means it shows make its copy. DNA has the ability to show its replication. DNA shows a replication process. It means DNA has the ability to make its copy. RNA also has the ability to replicate. It also has the property or has the ability to replicate. So on this difference, we cannot differentiate which one is the genetic material. There are some similarities and dissimilarities. This is the similarities. Next one is mutation. One word is variation and other one is mutation. Variation, two offspring produced from single parents and they have difference. That is called variation. Mutation, change in DNA which may cause variation, which may cause large difference. That is mutation. Mutation, DNA shows stable mutation and that stable mutation is essential for the evolution. DNA show the stable mutation. Which is essential for evolution. Means it may cause difference, but that difference is not so high that does not represent the evolution. DNA show also mutation, but stable mutation. RNA not show the stable mutation. The difference between the offspring may be large case suddenly. RNA does not show stable mutation. Why the RNA doesn't show the stable mutation? This is because RNA has more functional group than the DNA. And the compound which has more functional group, that is more reactive. More reactive is disabled. First is the reason which represent that RNA is the not genetic material. RNA has two functional groups. Which one? Hydroxyl functional group. whereas DNA has only one. RNA has two functional groups, and DNA has one OH group. The more functional group compound is more reactive and easily degenerate. It has more functional group, so it is reactive. So it is easily degenerate, not so reactive because less functional. Second one, this is the major reason of the mutation. Second one, DNA has adenine. DNA bonded two hydrogen bond with thymine to adenine. Or you can say in DNA, thymine, which is bonded with the two hydrogen bond adenine. So we can say that DNA contains thymine, which is double hydrogen bonded with adenine. But in RNA, in place of thymine, it contains uracil. It means adenine bonded with two hydrogen bond with uracil. And uracil is not stability. Uracil doesn't show stability as thymine stable. How is the uracil formed? Uracil is formed by the deamination of cytosine. Cytosine deamination, or we can say that cytosine by the deamination process change into uracil. So the percentage amount of DNA in cytosine may be reduced in nitrogen base, which may cause mutation, large scale mutation. So the RNA is not stable. Why the RNA show mutation? This is the first reason. RNA has two functional group, and we know that the compound which has more functional group, that is more reactive. More reactive compound easily degenerate. Second one is, in place of thymine in RNA, adenine bonded with two hydrogen bond with uracil. Uracil doesn't show stability. Why? Because uracil is produced from the deamination process of cytosine. If the uracil is produced from the cytosine, the percentage value of cytosine may be reduced in the DNA. So RNA is not the genetic material. One more difference, that is number third. The first one, second one, number third, expression of information. Expression of information. In DNA, or the information will be sent from DNA to mRNA and mRNA to protein, which is produced by the two different process, transcription and translation. That is called central dogma. How the information will be sent out in DNA as well as in RNA. As we have studied about central dogma, it means DNA is the genetic material DNA. This DNA change into mRNA. If the genetic material is DNA, the information sent out from DNA to mRNA, and the process in which DNA change into mRNA, that is called transcription. And then mRNA to protein. mRNA to protein. The process in which mRNA, messenger RNA change into protein, that is called translation. So the information, expression of information we send out, this process is known as central dogma. If the genetic material DNA, then DNA change into mRNA, mRNA to protein. This is called central dogma. If this process will happen, that is the genetic material. But if we compare it comparison the RNA with DNA. As the RNA, as any cell contain RNA, this RNA change into protein. Directly change into protein. There is no transcription process can take place. Only translation can take place. So the information not be sent out completely. This is the first thing. Second one, if the RNA is the genetic material, if any cell contain RNA genetic material, first one, the cell which contain RNA is the genetic material, that is called retrovirus or virus. If any virus contain RNA is the genetic material, this RNA change into DNA, and DNA again change into mRNA, and mRNA change into protein. This process may be completed as this one. But here we can see that RNA change into DNA. We have already studied what DNA change into mRNA or RNA. That is known as transcription. But this process is reverse. So it is called reverse transcription. Reverse transcription. Because RNA change into DNA. And this DNA to mRNA transcription. And mRNA to protein translation. So the expression of information in RNA may be reversed in this process. It is called reverse central dogma. On behalf of these differences, such like as replication, mutation, expression of information, as well as we can say that this one, the another part, nitrogen base. As we have seen that in DNA, adenine bonded with 2 hydrogen bond with thymine, but in RNA it bonded with uracil, and uracil formed by the deadenation process of cytosine. If these processes may be happened, it may cause large mutation, and large mutation is not essential or responsible for evolution. So we can say that RNA is not genetic material, whereas DNA is the stability. DNA shows stability, so it is the genetic material. In next part we will discuss on RNA world, and after that replication process.